So we finished talking about the maternal genes. Now we're going to continue with the following gene, that is the gap genes. Okay, so let's go back to this diagram that we have been talking, and I want that you observe what is the thing that grab more your attention. So when you start here, how many sections do you see? Yeah, you see anterior and posterior. And then you start going down here and you see, okay, there is three, four, five. Here there are more. Here there is more. Yeah. So what it's showing you is that as time goes by, the genes that are expressed divide the segment into much more complex shapes. Yeah. And this is what we're going to see. However, there will be a lot of feedback, a lot of hierarchy in this sequential pathway. And we're going to talk about that. We just finished the maternal gene effect. Now, here they are going to be the genes that are in the zygote, but those depend of the previous setup. So the maternal gene set up everything to go here, and now the zygotic genes are ready to start working. And I'm going to show you all this in the beginning from a general perspective, and then we will move at the end of the class to the genetic network from a very general perspective of how this works. Again, from a very general perspective, we're going to see some genes here. So from the gap genes, you have cripple, from the pair rule, Fusiteratsu, and from the segment polarity in gray. And in this diagram, you will see how the location in the embryo. Yeah, so I want that you see how they are located. Yeah, so again, I want to show you the general aspect. General aspect is that if you see, they now every time they start getting into smaller and smaller regions, yeah, more complex behaviors. And if you see where they are located, where they are expressed in the embryo, this one it was expressed in an entire region. Now this one will be expressed in stripes, and this one will be also expressed in smaller stripes. Yeah, so more and more complex patterns. In the larvae, this will be affecting an entire region here. And when it's lost, look what happened. Only the posterior region is here and the anterior is lost. Yeah. While in this one, what you're missing is entire stripes. So although it's smaller, you're missing this region. Something here is very similar happened here, but at a smaller scale. The stripes that are lost are even smaller. So what happened here is that you will see a regulator network that relied on feedback interaction and feed-forward interaction. Yeah, so what is determined, what happened here will determine what happened here, and then this one will determine what happened here. Yeah, and I'm going to show you at the end of the class the network and how to analyze that network for the test. But let's talk about first, from a very general perspective, what are these morphogenetic gradients and how they can create these segments, in particular these very tiny areas where there is a expression. So I want that you observe this diagram, and here in this diagram they show the cell fate of different cells. So you see some in green, some in blues, some in white, and some in red. And here you see a concentration of the morphogene that on the top is going to produce one type of cell. A lower concentration is going to have a different color of cell that represent a different cell, and so on. So here, what is showing is a morphogene, that is a signaling factor that directs the cell fate of a tissue and the distance from their source. In this case, we are showing only one molecule. In Drosophila, there are multiple signaling molecules that allow us to create more patterns. So here there will be one, another one they could be here, another one it could be here, that it can create even as another color in the cell, and another one could be here from a different gene. And this is how all these specific cell patterns appear. This one will be a different cell, and I will show you that. The best study system understanding morphogenes is in Drosophila wing. So is DPP, winglet that is very important, and hedgehog. Um, and they have been shown to work like this, but also in a three-dimensional pattern too. In this diagram, you can see two potential mechanisms for the morphogene. Yeah, there are several potential mechanisms. Here you can see the one for wingless and here the one for DPP. So the one for wingless is the diffusion through extracellular space. So in this case, it could be something like a paracrine signaling that the molecules go outside, and there will be a gradient. The ones that are closer to the molecule, the morphogen source, will have a higher amount of the signaling, while the ones that are farther will have slowly less and less. Another one is the one proposed for DPP, that is actually transcytosis. Instead of the cells going outside, they go between cells through vesicles that go from one to another one. But of course, there will be less molecule the farther that the cells that are, the lower the number of molecules that there will be. So remember, diffusion through an extracellular space, like in winglets, and planar transcytosis, like in DPP. So, and in Drosophila, there is a different mechanism. Why? Because hopefully this you remember what is a syncytial blastoderm. That you remember there are no cells. The cellular blastoderm occur later between 13 and 14 stage. And here, what is happening is that all these gap gene expressions, all these transcription factors, are diffusing freely among the nuclei. Yeah, and it allows us to establish a morphogenetic gradients of proteins, as well as domain of gene expression for the messenger RNA. Okay, so let me show you just a final diagram. So here you see a syncytial blastoderm, and here you see a cellularization. So is before around those time where maternal genes and gap genes are used, and so it's a different mechanism. Oh, besides the one that I talked before, the two different mechanisms. Yeah, and later during cellularization, the cells are going to I may use the other mechanisms that I described. Here you can see the fate map of the embryo, of the larvae, and of the adult. So maybe you remember that flies in adults have head parts, thorax, and abdomen. In the embryo, this in blue will be the head, the thorax will be this one, and in green you see the abdomen. These three areas, the two ends, are not segmented. They go to a process of invagination, or they migrate the cells to form internal structures of the head and the gut. If you remember the pole cells, they form the hindgut here. In addition to that, the segmentation can be divided in two terms, segments and parasegments. So the parasegment, if you see, remember there are 14 parasegments. Those one are the one that we find in the larvae and also at the end of the embryo. Yeah, that we count each one of those, and the segments are the one that we do for the adult. However, if you go beyond the phenotype and you go to the genotype and to the development, you realize the parasegment actually correspond to the patterns of gene expression. Yeah, to the cascade of events, yeah, the produce these developmental units that are the parasegments. In particular, they are narrow stripes, so you see denticle space between the wingless and ingrained patterns. Again, I'm not going to ask you to memorize all the genes, but for the pair rule genes, this is one that is very important to remember. Even skip, also Fushiteratsu for the pair rule genes, and also for the segment polarity in Grail, wingless, as well as hedgehog. So what we have been talking is that when you move from a maternal gene to the homeotic genes, you start seeing more and more specificity in the each one of the segments. So the second level of specificity is these gap genes that they were found by deletion of several adjacent segments. So they said, okay, we're missing this segment again, we're missing this segment, we're missing this segment, and they start analyzing all of those. And then they start analyzing the patterns of expression. So now what I'm going to show you is several patterns of expressions using diagrams of some of the gap genes. Okay, now I want that you give a look to these different patterns of expression. I want that you see what you find in common. Yeah, so you find that they are, when you put all of them together, you start finding more and more segments. So for example, this one, nerves and tailless, they are like the opposite. They are like mirror images of what is happening. One goes to the anterior, the other one goes to the posterior. Hunchback, we already talked about it. But if you put all of them together, you see that they start forming more and more stripes. Yeah, so they start dividing up the anterior and the posterior length of the embryo into this gap gene of expression. And in the next slide, I'm going to show you an example of a mutation, showing you what happened if you remove them. And the same will happen, the same type of experiments they did with that. And then I'm going to show you the genetic network of how all of them regulate each other. So don't get anxious, just don't you need to memorize the genetic network. I just want that you know how to read it. So let's start. So in this slide, I just want to tell you that the gap mutants lack a large region of the larval body. So here you see the expression of crupple that is in the medial region. You can see it here in this diagram, as well as in the embryo here in this part of the diagram. And in the normal larva, crupple will be located here. However, when you have a mutant, the mutant will be lethal. The fly is going to die and is going to miss all these segments here. So this is how it was studied. Each one of these gap genes were they were missing some sections, as we said before, and they then later they were studied in which part of the embryo was located. So now let's go and try to imagine that I'm not going to show you the same picture for all the genes. I'm just going to show you, because it is very logical what they did. Now I'm going to show you how they create the network to understand the development. Okay, so here is the entire network, and they use all this information to create all this network of interactions. And I'm going to show you how to read it. Yeah, using Bicoid. Remember, the arrows indicate positive regulation, while this symbol means inhibition. So let's start with Bicoid. Here you see it very clearly. When you go down here, you see that it positively regulates this gene that is in green. Here and here it does the same thing. On the other hand, if you see Bicoid, you remember it will inhibit caudal. Yeah, and the same thing, you can do it with other genes. So for example, the torso, yeah, it will positively regulate these two genes. Giant, if you see the gene giant is positively regulated by Bicoid, but it will be negative regulated by this one in red, the green one that is tailless, yeah, and in that way you put all the information there. In the test, I'm not going to ask you to memorize all the names of those. I'm just going to tell you, okay, a Bicoid is positively, I will give you the names, yeah, and you just have to tell me if it's positively regulated or if it's inhibiting. Okay, so it's pretty simple, and this is how you read these genetic networks. And what happened here is that you will have a network, a hierarchy of interactions. So for example, here if Bicoid is here positively regulating hunchback, here it will hunchback, it will be repressed here, as you see by this symbol in purple. and it will cripple, it will be positively regulated. However, if you have a mutation here in bicoid, hunchback is not be able to have the action in these genes here. It will not be able to repress or positively regulate. So as you can see, there is a feedback of the interactions as we talked in the beginning. Hopefully now it's clear why is cripple have these beautiful bands, because it has a relationship with these genes, with hunchback and cuddle. And we now know that hunchback here, when it's at high levels, it will repress cripple. However, at intermediate levels, yeah, the bands might be different. Yeah, they will, instead of repressing, there will be an activation. And this is just one example that I took. Same thing will happen with other genes. It's just as an example so you understand how this works. And now try to answer this question that is pretty easy. Refer to the diagram that is shown below to respond the following question about the gap genes caudal. So you see the gene caudal here, which genes have their expression regulated by caudal. Is caudal gene regulating those genes positively or negatively? So pretty easy. You see caudal here, and it's regulating positively both nerves and giant, or yeah, nerves and giant. Let me put it so in red, and here you see the arrow here, and here you see the arrow. Good. So pretty easy. So far we have talked about the maternal genes, the gap genes, and now we're going to talk about the pair rule genes, and we're going to start just with a small summary of what we have talked so far. In this table, they summarize what we have talked. Here is the genetic hierarchy. Here are the function of those genes and the representative genes that you need to memorize all of them, but I'm sure that you now remember some of them, and the effect of the mutations. So the first group is the maternal gene effect, and here this one, what they do, the function is, remember, to know what is the anterior and the posterior, the major sections, as well as the terminal regions. Yeah, that we talk. Now I'm sure that you remember genes like decoy, like Oscar, like caudal, torso, all those things. And you know that when there is a mutation, there will be a major disruption in the organization. You might end up creating two posterior, a larvae with two posterior regions, or you might end up creating a major disruption in the morphogenesis. Then we start talking about segmentation genes, and we start with the gap genes. So now we start dividing in more detail in the body into different regions. Yeah, into broad regions. And here are the genes like purple, and we start finding how you start forming all different regions. And when you have a mutation, you are missing a big portion of the segment, like what we studied there, and with cripple. And now we're going to move into the pair rule genes that now we're not going to define broad regions. We're going to define much thinner regions. We're going to define 7 segments. And I told you that, remember, even skipped and Fushi Teratsu. And instead of the previous effect that is going to create major changes in the anterior posterior or in major areas of the embryo, here you're going to find that you delete every other segment when you find a mutation, for example, in Fusiteratsu or in even skip and other genes. In previous stages, you have divided the body in big portions, yeah, in broad portions. Here is the first time that you see segments, yeah, blue segments, gray segments, yeah, and this will continue and will be responsible for creating the segment polarity. And later, if you remember, with the parasegments of the larvae that we were studying, the same thing happened here. So all that start with the para-rule genes. Here in the diagram, you see how you go from the gap gene expression to the pair rule gene expression. So slowly you start finding that stripes start forming, yeah. And I'm going to show you how that happened, how the genes again are interacting through morphogenes start creating these stripes. And at the end you see very clear stripes. And here the mutants are also going to get small, yeah, but they are going to get small in a different way. They start missing now specific portions, yeah, of the segments. So let me show you what happened. And to do that, I wanted, instead of showing you, I just want that you think. Based on this figure, what is the expression pattern of the gene Fusiteratsu? So here you have the wild type and you see the stripes. How do you see those stripes? So here there's one stripe, yeah, in the normal, and here you see another stripe, and here you see another stripe, yeah, and here you see another stripe. And now I want that you see these stripes. Do you see any in this stripe? What do you see in the following stripe and in this following stripe? Is okay, so here you can see staining of the genes even skipped and Fushiteratsu, and as you saw in the previous slide, the normal stripes will be much thinner. If even skip is gone, what you're going to find is the stripe much wider. And as we have said multiple times, here is the first one, and if you count, there will be 7 stripes. So you are going to have half of the segments. So here you see that here there is A7, there is only 7 segments. And just to finish, the generation of this pattern of segmentation and gene expression patterns coincide with the formation of the cell membranes around the blastoderm. So we don't have any more desyncytium. At this point, we already have all the nucleus and cell membranes there. This following slide, I want that you think, how does the gene expression stripes are forming the Drosophila embryo? So if you see here, this is the gap genes, and at the end we have the pair rule genes. And it's in B and in C you see that they start fuzzy and they start becoming sharper and sharper. And the question is how this happened. And there are 4 genes that I want that you analyze here. Yeah, so bicode, giant, hunchback, and cripple. What do you think that is the interaction of this gene is allowing the formation of this if stripe 2? Yeah, we're not going to go through the 7 one. I'm going to use number 2 so you can understand how all of them are formed. Okay, based on this diagram, what are the genes that are positively and negatively regulate even a strip? I magnify, zoom in into the figure so you can see it better. Here is the stripe number 2, and here are these genes. So which one do you think that are negatively regulated, and which one are positively regulating this parasegment number 2? So how we find the answer? The first thing that we need to know is, okay, where is this gene located? Yeah, in the parasegment tree, and what is happening with the location? What other genes? What is happening with the shape and position of the cytoplasmatic polarity and the gap morphogenetic gradient? So we can see, for example, that picoid is expressed there. Yeah, we see hunchback is also expressed there. However, you see that giant and cripple set like borders. Yeah, so it seems that giant is a negative regulator of the anterior border, while cripple is a negative regulator of the posterior border. The positive regulator could be hunchback or bicoid or both. And this is how this if strip 2 can be formed. And something similar can be analyzed when you analyze each one of the other strips. Okay, so with this we finished the pair rule gene, and I'm going to play a video that summarizes everything from here up to here. Hopefully it makes life easier for you, and if you have any question, just let me know. Have a good day and bye.
In Drosophila embryogenesis, the body plan along the anterior-posterior axis is determined by a cascade of transcription regulatory events. Maternal mRNAs such as bicoid are required for early patterning. Bicoid mRNA is localized to the anterior pole. Once synthesized, bicoid protein diffuses posteriorly, forming a protein gradient. Bicoid protein activates transcription of the hunchback gene by binding its promoter. The expressed hunchback protein is localized to the same region as bicoid. The hunchback protein acts as a transcriptional activator or repressor of several other genes, including cruple. At high concentrations, hunchback represses cruple transcription. Below a critical threshold concentration, hunchback activates cruple. At a second lower threshold concentration, hunchback no longer activates cruple. Thus, cruple protein localization is restricted to a central band of the embryo. Bicoid, hunchback, cruple, and a fourth protein, giant, control expression of even-skipped stripe 2. Bicoid and hunchback activate transcription of the even-skip gene in a broad band, while giant and cruple repress its transcription at sharp anterior and posterior boundaries. This generates a precise stripe of even-skipped protein. Stripe 2 is one of 7 stripes of even-skipped protein, which alternate with 7 stripes of Fushi-Toratsu protein. Both proteins are transcription factors encoded by pair rule genes. Pair rule genes, along with segment polarity genes, define 14 parasegments of the embryo. Selector genes regulate development within each parasegment. This determines the corresponding adult body structures.